Well, yeah. So is this on? That's on.
Yeah.
Okay. Great. Well, thanks so much for Robin and Ade for generous introductions. It's a pleasure to be back here. It's not always like this in Davis, but this in the springtime, the botanical gardens there in Arboretum are really nice. So this is the cardiac myocyte, and probably you've all seen a version of this from the EC coupling standpoint. And I was here last year for the UCARS symposium and talked all about adrenergic signaling and PKA. And today I'm going to focus more on CAMP kinase in the context of the diseases that I'm talking about. And I'm going to start building up from some old stuff. I'll try to go through quickly into some of the most recent stuff. So CAMP kinase, like PKA, phosphorylates a lot of targets in the myocyte. In all different regions, including in the nucleus for transcriptional regulation. And everything on this slide we study one way or another. And trying to, again, incorporate into quantitative models to test our understanding of things. And CAMP kinase can also move around. So let me tell you a little bit about CAMP kinase targets in more detail. So CAMP kinase phosphorylates the rhamine receptor and phospholamban. And on the RYR, it creates CPVT-like modifications in the behavior. For those watching online, I apologize. I'm using a laser pointer on the screen, but hopefully the images are clear enough. The rhamine receptor, then, if it's overactivated, can release calcium and lead to delayed after depolarization, which if sufficient can trigger an action potential or PDC. It also modifies. What's this for? Field infection. Modifies the gating properties of L-type calcium channels to prolong the activated state, and therefore can contribute to early after depolarizations as depicted here, with longer action potential durations. But also phosphorylation of the sodium channel modifies the gating of the sodium channel in ways, complex ways that both can look like LQT3 or Brugada syndrome, depending on hardware. And in fact, mutations in the sodium channel mimic exactly that scenario. All these K channels are modulated both acutely and in some cases transcriptionally by CAMP kinase regulation. And these K channels can contribute to an increased dispersion of repolarization, enhancing the reentrant substrate in the myocardium. CAMP kinase also has effects on contractile proteins. I won't talk much about that today. And as I mentioned before, on transcriptional regulation through HDAC and other pathways in the nucleus, which I'm not going to talk about really at all today. But I will say that we used to think of CAMP kinase as getting activated only when calcium went up, like after beta adrenergic stimulation or so forth. But it's not really that simple. Indeed, over the past 10, 15 years, NOS, ROS, and glycinac, which I'll have more to say about, a sugar molecule, can covalently bind to CAMP kinase and modulate and promote its activation state. So it becomes a really complicated signal, looking like a PKA signaling diagram from 10, 15 years ago. But we're starting to understand how some of these pieces work. And to start with, this should be a slide I go at the end with, but since you'll probably fall asleep halfway through this talk, I wanted to introduce this one here. So in heart failure, we know that there's a leaky ryanodine receptor. So, and that's true in CPVT as well. In heart failure, CAMP kinase is also upregulated and activated, and we're talking about HFpEF here in particular. And we know that that elevated CAMP kinase can regulate the RYR to worsen the leak, promoting more CAMP kinase. So there's a positive feedback cycle here. And that kind of extends broadly in the cell, such that it can promote the incidence of reagents and arrhythmias. That CAMP kinase can also phosphorylate and regulate sodium channels, as I mentioned, and can lead to longer action potentials and increased sodium entry via late current. And that sodium elevation can also promote further calcium increase. But it also has an interesting point where sodium-dependent calcium extrusion from mitochondria can lower mitochondrial calcium, which can increase ROCK production. And ROCK production is actually also able to modulate some of these same targets. And so there's a feedback cycle here, and where ROCK can also activate CHEM kinase. So this positive feedback loop, which is a vicious cycle, we think, gets engaged in heart failure. And after CHEM kinase gets activated, it can translocate and affect other targets, including nuclear transcription, which I mentioned. So because this is kind of a vicious cycle, you may not have to block the whole thing. You might be able to break the cycle in key points. And we'll talk about some of the possibilities about that later. But I would propose and talk a little bit more about that this same pathway of vicious cycle can get activated in both diabetes acutely, acute hyperglycemia, and in HFpEF, which I will talk about more at the end. So a quick primer on CHEM kinase. It's a dodecamer structure, and each monomer has a regulatory, catalytic domain, and an association domain. It normally sits in an inactivated auto-inhibited state when the regulatory inhibits the catalytic domain. But when calcium calmodulin comes along, it opens it up so that the catalytic domain can phosphorylate targets. One of the key targets is the neighboring subunit in this dodecamer, where if it's phosphorylated at this known site of 287 and CHEM kinase delta, that promotes the autonomously active state. So even when calcium calmodulin comes off, it stays active for a period of time. And that was well described many years ago with CHEM kinase. But what's come to be clear in more recent years, and I call them the Erickson sites, because Jeff Erickson is the one who identified all these different nitrosylation, oxidation, and gluten acylation sites from CHEM kinase that promote that same kind of autonomous activity. So I'll take you sort of backwards here to a question that came up where there are intrinsic mechanisms of cardiac autoregulation. It's kind of a side story, but bear with me. So we all know about the Frank Starling effect, where you increase the sarcomere length, and immediately there's an increase in the strength of contraction. So that's very familiar, I think, to all of us in learning about how the heart autoregulates. Another way that the heart can autoregulate is when there's an elevated afterload. And that's often referred to as the anaerobic effect, or slow force response, where gradually the force builds up after the stretch. And it's creating an afterload, and that is a calcium-dependent increase in force development here without changing sarcomere length. Now, in our department, Yi-Chen Mizu has developed a cell-in-gel approach where you can tune the stiffness of a three-dimensional gel. And what I liked about this was because it allowed us to separately look at what afterload does, because the cells are kind of locked into their resting length. And then we can change the stiffness of the gel against which the cell has to contract. And so what Yi-Chen Mizu reported in his first paper a number of years ago now was that if you put this same cell in a gel, it doesn't shorten as much because it's developing force. Not a surprise. But what was a little bit of a surprise was how big the calcium transient got. So it increased the calcium transient substantially. And it also promoted calcium sparks that you can see in this sort of 3D plot up here. And in NOS1 knockout mice, those sparks went away and the amplitude of the calcium transient effect was suppressed. And when she put a CHEM kinase inhibitor on, that also suppressed it. So it looked like this slow force response may require both nNOS, so NOS1, and is prevented by CHEM kinase inhibitions. So that was going on at the same time Jeff Erickson, who I already mentioned, who's now an associate professor in New Zealand, was studying nitric oxide as a possible modulator of CHEM kinase signaling. And this is a snapshot from this JVC paper from then, where if you put calcium, this is looking at this Kamui reporter that we used. And you can see that if you put calcium on, it doesn't do anything. If you add calcium and calmodulin, it activates. You take away the calcium, it comes back down. If instead you do the activation in one step here, and then you put on G-SNO to S-nitrosylate things, what you see is the CAM kinase stays activated, the autonomous activation we talked about. And a single site mutation on CAM kinase is sufficient to completely prevent that autonomous active state. So this C290 site that's right next to where calmodulin binds on the CAM kinase is sufficient to promote that effect. So a graduate student in the lab later, once we had the mice all described here, we reproduced those fundamental results of the afterload increasing calcium transient and also speeding the rate of calcium decline. So Dira did the same experiments in CAM kinase cardiac specific knockout mice, and there was no longer an increase in the calcium transient and no acceleration of calcium decline. Then she also did it in new mice that we had developed that lacked that single cysteine nitrosylation site. And that also completely abolished the tau and the increase in calcium transient. So it seems like that calcium or that CAM kinase site at 290 is essential for afterload induced increase in calcium and SR uptake. Indeed, the calcium sparks effect was also abolished in the knockouts or in these point mutations. So it looks like then there's mechanical afterload, and Dr. Izu's lab is now looking more at the mechanisms, how that mechanical load introduces this increase in NOS1 activity. But clearly that's nitrosylated in CAM kinase, and that's phosphorylated RYR and some other targets. So there's another interesting thing happened when Jeff, everything is about CAM kinase with Jeff, so it was good to have in the lab. And Leticia Pereira, a French postdoc, came to the lab and she said, well, I put the cells in high glucose and they get way more sparky. So a lot more spontaneous calcium releases. And so Jeff said, it must be CAM kinase. So he looked at the CAM kinase activity at higher glucose outside the cell, and he found indeed higher glucose promoted CAM kinase activity. And it was exacerbated by co-activating with isoproterenol. So that happened to be at the same time I was trying to recruit Jerry Hartford, who is the GLICNAC expert of the world, I think, from Johns Hopkins to UC Davis. And he said, and I was talking to him, and he said, well, yeah, maybe it's like this, because a lot of proteins get GLICNACylated at serines and threonines. And so it's an interesting additional post-translational modification other than phosphorylation, oxidation, and phosphorylation. And so we explored it, and what we found out was indeed there was GLICNACylation happening at this serine here, right next to the oxidation sites in this very busy regulatory domain. And so, and testing that, what Jeff did was to say, well, okay, let's inhibit the one enzyme that puts the GLICNAC groups on, and that abolished the response. And let's try inhibiting the enzyme that takes it off, and that promotes the response. So it's pretty clear GLICNACylation is involved, and this mutation at this site prevented it in vitro in this paper. And so high glucose promotes calcium sparks, as you see here, and inhibiting either CAM kinase or GLICNACylation prevents that increase in sparks. So it looks like this site is quite important for that. And if you knock out CAM kinase delta, which we had mice for at that time, you could also abolish the effect of inhibiting the removal enzyme Thiamine G, which created hugely sparky myocytes. So Crystal Ripplinger was in the department then, and we asked her, or maybe the Nature editors asked us to show something in a real organ. And so she did these optical mapping experiments with a crushed PV and showed that just high glucose promoted these PVCs to occur. And that was inhibited by Don. That's not just me coming in the room, so don't do it. But it's that inhibitor of the GLICNACylation process, or by inhibiting CAM kinase. So the interesting thing too was she happened to see some incidents where the calcium rose before the voltage in these dual mapping studies, which is a kind of a hallmark of a calcium-induced PVC. We also looked in human samples from heart failure and heart failure with diabetes patients, and glutination of CAM kinase was elevated. That was also true in human brains from diabetic patients, and also true in a more controlled model, animal model of diabetes. So it looks like this high glucose promotes this pathway toward glutination of CAM kinase and phosphorylation and consequences with downstream targets of CAM kinase, which could include more of the ones than I talked about before. We also came across a paper that suggested high glucose could also promote ROS production, and that it might be CAM kinase dependent, but there was no explanation of how that might occur. So a graduate student in the lab sought to test that. So using both DCF and data, I won't show you a genetically encoded ROS reporter, which showed kind of the same results, was that you would see an increase, about 40% increase in ROS production just by changing glucose to a high hyperglycemic diabetic level in the cell, outside the cells. And that effect was again ablated by either inhibiting CAM kinase with KM93 or now the glutination with a better enzyme. And this just shows that thiamine G, which inhibits the deglutination, by itself was sufficient to reproduce this effect of high glucose. And the remarkable thing was, okay, there's more ROS, but contractions weren't changed, relaxation wasn't changed, calcium changes were hardly changed. But the thing that stood out was there's a big increase in calcium sparks, and again, they're suppressed by these inhibitors. And what Shan went on to show was that in CAM kinase global knockouts, cardiac-specific knockouts, that effect was completely abolished. Also, the calcium spark subsequent effect. So this seems to be important for both the ROS and the RYR kind of effects. And that if we use the point mutation CAM kinase delta that deletes that site, the effect is abolished. We tested that if we try another way to increase ROS, it still works. So they can't not make ROS. But this pathway of hyperglycemia-induced ROS production was blocked. And it was also blocked in NOX2 knockout mice. So we couldn't see that increase anymore. So it came together, I'll skip over that the idea is that glucose can promote the glutination of CAM kinase again, and that could promote NOX2 in the plasma membrane to produce ROS, and that ROS can then do other things through ion channels, et cetera. And what we found, we expected to see a big feedback here, so another vicious cycle here going this way. But we saw the experimental data with other mutants suggested that was a minor effect. The ROS production was really driven more directly by CAM kinase. So Jeff is back again when he went back and went to New Zealand. He had access to some human samples, well-matched non-diabetics and diabetic samples, and found again that in human diabetics, that there was an elevation in glutination. But the other site that he had previously identified, the oxidation sites, were not really changed in the diabetic patients. So to study kind of the chronic and a little bit more on the acute effects, we took advantage of those knock-in mice that we made with that point mutation. So the good news is at baseline, they have virtually no phenotype. I'm not going to go through this whole table. And Ben Steggy and Chris Coe published this paper looking first at high glucose. And if you put high glucose on in normal myocytes, again, you don't see much change in the calcium transient. As I mentioned before, you see big increases in calcium sparks, as shown here, and those are ablated in the absence of delta CAM kinase or when that one point mutation was done. So for some real action potentials, then it also shows here that acute increase in glucose, so this is going from 5.5 to 30 millimolar glucose. We wanted to get a full effect. That increases the action potential duration and also reveals cardiac alternans, APD alternans out here at pacing rates that are actually relevant for a mouse. And those effects were both completely gone in the S288 knock-in mice. Not surprisingly, they were also gone in the the cardiac-specific knockout of CHEM kinase, but they were still present in the oxidation-resistant CHEM kinase mice. So the oxidation site wasn't critical for this. It might play an additive role, but it wasn't critical for it. That was also true for these DADs that were measured in the same kind of experiments with fast pacing and stopping and looking for spontaneous calcium releases, in this case, that can lead to a full-blown action potential. We didn't see that increase in the S2A mice. But then we wanted to stress them with kind of a more chronic stress with a type 1 diabetes model, this case with a multiple small STZ, streptozotocin injections. They developed, robust hyperglycemia, normal ejection fraction. They were more arrhythmogenic at the whole heart level, and they had a little diastolic dysfunction. Well, I'll come back to diastolic dysfunction later. Again, the calcium transients, not much different. I won't go through all these graphs, but the thing that stood out was you see more of these propagating calcium waves in the myocytes, which are the things that trigger the transient inward current that is producing DAD. And we looked at calcium sparks, and even the streptozotocin myocytes, when we take them out, isolate the cells, do these kind of experiments, so they're not in high glucose anymore, but they already had twice calcium spark activity. And if you put them back at the level that they have circulating glucose, the cells have this robust increase in calcium spark frequency. And once again, this 1 point mutation in CHEM kinase fully ablated it, and the oxidation maybe partially ablated it. So there might be this crosstalk between these two PTMs on the CHEM kinase activation state. Looking at the APs, I'll go through this quickly because when they're in chronic hyperglycemia, everything's just worse, but the same stuff happens. You can see alternans here happening at a much lower frequency. The APDs are even longer in these mice when you elevate glucose. And again, that's almost completely prevented in these particular point mutation mice here. What was really striking too was the DADs in these chronic STZ mice were really substantial. And when you go back to the calcium or the glucose concentration that they were living in the heart, wow, that's a lot. It's hard to get that many under any conditions. But what we found in the knocked-in mice was, no, they don't care. So this pathway seems to be really, the CHEM kinase at that one site seems to be really critical for this high glucose induced pathway for all the stuff that I've been showing you. We've also looked at other stuff I won't have time to go through on late sodium current, potassium channels of various sorts, and that's published in some other papers here, but I don't have time to go through that today. I want to get to the HFpEF. And so to summarize this part, an acute hypoglycemia increases ROS production, calcium leak, reduces calcium load, increases APD, alternans, and arrhythmogenic DADs. And all of those seem to be importantly suppressed by this one site on CHEM kinase. In diabetes, everything just gets worse. And there's probably a chronic part of this that may have to do with CHEM kinase's effect on transcription and altered gene expression. So it's not all immediately reversible when you isolate the cell. It's still sensitive to acute hyperglycemia. So I'm going to move from that background, and I'll get to talking about HFpEF. So we're all familiar with HFrEF, and we've studied that and mechanisms that are important in cardiac dysfunction and HFrEF before. But as all of you know, HFrEF is becoming an increasingly, and more than half of the patients diagnosed with heart failure are HFpEF patients. And a lot more of them tend to be diabetic or obese. So there's been kind of a shift in the HFpEF population from what used to be a thin, hypertensive female patient to a more broad cardiometabolic, obese, diabetic patient who exhibits HFpEF symptoms. So Joe Hill's lab developed a high-fat diet in Elney model, which produced very nice diastolic dysfunction with preserved ejection fraction. And it had some hypertrophy, some pulmonary edema, a little obesity, not really hyperglycemia. But we thought it would be nice to have a complementary model that really emphasized this new, more obese diabetic model. And Ben's Heggie in our groups really developed this model with a lot of help from Juliana Mera Hernandez and Chris Coe in the lab. And so indeed, actually a bigger increase in diastolic dysfunction from EO3 prime. Hypertrophy and lung weight also went up like with the Hill model. But here, obesity and diabetes were much more apparent. And if we look more closely at this, the ejection fraction is pretty normal. And notably, if you just do aldosterone alone or just have EVDB mice by themselves, they don't develop this HFpEF phenotype. But you put them together and you start to see left ventricular LVRI median increase. And then if you look at the E over A or E over E prime, you see a robust HFpEF phenotype. And there's also an increase in left atrial volume. So the combination seems important. So we compared some fundamental properties of these and compared to the Hill, I call it kind of hypertensive versus a volume overload. The aldosterone creates a little volume overload on top of the hyperglycemia in the DD mice. And this new model then has kind of more robust indicators of HFpEF phenotype. And one of the things that we found really interesting that I'll follow up on here is that in the Hill model, the females are generally protected. And in our model, the females are worse. And we wanted to figure out why. So there's also a big difference in the overall gene expression changes in the Hill model here in the pink columns and our DBD-ALDO model here. And that goes for adrenergic signaling, big differences between the models, and also with calcium handling signaling. So indeed, the calcium transients decline more slowly. So calcium transients have longer taus. And that also results with pacing with an increase in the diastolic between beats level of calcium, which of course that higher calcium level between beats could be diastolic dysfunction because the cells may not and the heart may not relax as well. I'll come back to that in a few slides, but I mentioned empagliflozin in the title and I thought I'd bring it up here because when we started doing these models, both the Hill model and our new model, one of the things Ben was keen on testing was the late current here. So in both of these models, you can see there's an increase in the late current that's kind of plateaued out here for sodium current. And TTX, of course, abolishes it, but EMPA also reduces it back to the normal level. And you can see the data down here. What's interesting too is it doesn't, in our hands, it doesn't seem to be a direct effect. You have to pre-incubate for at least half an hour. In this case, Ben pre-incubated for 4 hours with empagliflozin to obtain these clear results. And this is the same kind of data in the Joe Hill model where there is an elevation in late current and it's also inhibited by empagliflozin. Whoa. That's interesting. Yes, I have a slide number here. Oh, I was close. So that late current also we think contributes to the longer action potential duration, and EMPA also reduced APD back to the control level. And the same was true for short-term variability of action potential duration, which is another really interesting readout that I think is underutilized. It also reduced that. So there was a more stable beat-to-beat variability for APD. And we did see, again, same pattern in the Joe Hill model with respect to both APD and STD. Interestingly, in this series, we also inhibited CAM kinase with a peptide inhibitor that's very specific, and it produced the same effect. So our current thinking about EMPA is these acute effects are mediated probably upstream of CAM kinase, maybe with involving ROS as well. but inhibiting those can protect the late current from developing at least to that same level. So all these post-translational modifications are really important. The same thing was seen with these DADs. They were increased in the DBD-algo model, but they were suppressed by acute treatment with empagliflozin. So now I'm going to talk about sex differences. So it was sex in the title 2. That wasn't just to get a bigger crowd here. But what we saw was that in the female mice, that the increase in diastolic dysfunction was more pronounced, whether in either of these measures. And so we wondered if we could track down what's going on. And this again was Ben and Viviana and Aaron, both of who are BBMs actually too. So now, if I go back to that calcium transient decline slide from before, and we break it down into male and female, what we find is that the calcium decline and diastolic calcium are elevated in male, but not female. So here, the calcium decay gets slower, diastolic calcium is higher, significantly so in males, and there might be a slight trend in the females, but not nearly as much. So that data I showed you before was dominated by the males in our mixed population. So that means in the males, maybe the slower calcium decline is important and contributes, maybe not explains, but contributes to the diastolic dysfunction. Calcium just doesn't get down between beats. Heart doesn't relax as well. It's stiffer. But why is the HFpEF phenotype worse in the females? So we said, well, let's look downstream of calcium. And we looked at myofilament properties. And we looked at phosphorylation of troponin I that regulates myofilament calcium sensitivity. And what we found is that males and females go opposite directions in how they change during the development of HFpEF. And so a simple way to translate that is, so in the females, there may be normal diastolic calcium, but with the lower level of TNI phosphorylation, that moves this curve to the left. So you get higher force without having higher calcium. In the males, the higher force that you get there is potentially because of higher calcium. So it makes sense then from this standpoint that the myofilament sensitivity or stiffness and maybe other factors may be more important in the female. So we also looked at fibrosis and periostin as a readout, and that was a little more pronounced in the females than the males. And then we also got Pancratier to help us with analyzing Titin. And what he found was this phosphorylation site, which is a both PKC and CAM kinase phosphorylation site. It was up in the females, but not in males. So that would make the Titin stiffer. In addition, there was a slight but significant trend for an increase in the level of the stiffer isoform of Titin, this NB2. So our conclusion from that story is that in the males, my level of calcium seems to be at least partially sufficient to explain what's going on in the males in terms of calcium dysfunction. I didn't mention it before, but there's also decreased cell-type calcium current. So the calcium side in my wheelhouse seems to be more important in the males, whereas the myofilament properties and fibrosis, things not involving calcium, seems to be more important in females. So if this model of a diabetic obese mouse has anything to do with different classes of human patients that are obese and diabetic versus those with hypertension, this might mean then that the way we treat females with HFpEF might be appropriately different between males and females if they really have a foundational mechanism that's different. We don't know if that's the case. It's hard to, and I'll come back to some reasons later why. So we did another series of studies. So Ben Peggy was really industrious and reproduced the Hill model in all of these different CAM kinase mutant mice that we have, the knockouts, the cardiac specific knockouts, the 3 different mutants here. And what's interesting is we see this increase in E over E prime here in the wild types. If you take out CAM kinase completely, doesn't happen. So CAM kinase is also central to the development of the HFpEF phenotype. If we get rid of our glycylation site, the hyperglycemic induced one, doesn't matter, just like control. If we get rid of the NOS nitrosylation site at 290, doesn't matter. But if we take out the oxidation sites, they don't develop HFpEF. So in this case, it seems like for this particular Hill model of hypertensive HFpEF, the oxidation sites on CAM kinase seem to be critical. In contrast, and so far we only have this one genetic model to compare. In the DBDB-ALDO mice, you can see here the degree of diastolic dysfunction is either almost on the same scale here. You can see it's much more dramatic in the DBDB-ALDO model. But if we do that in the S280 non-glycosylated, it almost completely preserves function, so they don't develop it. This is the hyperglycemia, and we showed all the acute effects and chronic effects of hyperglycemia that are invoking this particular site. Now, these are tough because the DBDB mice are infertile, so getting the crosses to be both homozygous for the knock-in and for the DBDB is a pain. And so we're doing this now with the oxidation resistant mice to see if they are less protected here than they were in this model. Okay, so how about empagliflozin? I teased you before today. So we decided we did those acute experiments and showed pretty dramatic effects on isolated myocytes, which, as far as we know, do not express any SGLT2. So these are off-target effects. And so far, we think they're good off-target effects. And I think other people have found similar things in other contexts. But now we wanted to say, well, if we treat these mice, these very obese diabetic mice that have developed substantial HFpEF, can they get any benefit from in vivo treatment with empagliflozin at dosages that we think resemble what are used in patients? And what we found was that EMPA in vivo reduced the hyperglycemia. That's not surprising. It reduced the BNP, a readout of dysfunction there, and the lung weights were down. The diastolic dysfunction was reduced, not quite back to normal, but it was substantially reduced. And there was also a little reduction in the left atrial area. But not surprisingly, there was no effect, well, maybe not surprisingly, but there was no effect on body weight, LV mass, and of course, all of these have the same ejection fraction, which is kind of convenient if you want to talk about HFpEF. So we also did the same kind of electrophysiology experiments, and to make that quick, at baseline, the wild types that aren't in the protocol for HFpEF, HFpEF effects are pretty modest, nothing striking. But here, the in vivo treatment, even though now we isolate the myocytes, it takes a couple of hours, then they're cultured and be sitting around for another couple of hours while the experiments are going on. Even with no EMPA during that time, there's a lasting beneficial effect on the APD. It gets shortened. The alternans gets reduced. You can see here the EMPA is down here and it hardly splits until the very end. And short-term variability of APD, which is shown here. So all these things are significantly or substantially improved by EMPA treatment with vivo. Resting memory potential also becomes more negative. And consequentially, for those who know about sodium channels, that may explain why the DDDT max is faster, because the cells are probably more polarized, more sodium channels are available. It also improved these delayed after declarations back to kind of the control level here. And what I won't show, because I showed you enough data slides already, is that it also reversed the increases in the late current, even though it wasn't there during the actual measurement. So this is maybe a longer term effect, again, suggesting that maybe something upstream is influencing the remodeling of behavior, the regulation of that. It also reverse the decrease in IKS, calcium, and IK1. And the IK1 can also, when that's down, be kind of arrhythmogenic for lots of reasons the electrophysiologists will know, and also the slow calcium decline and transient decay. So almost everything that was creating them dysfunction was suppressed in this 4 weeks with the empagliflozin treatment. So we thought, okay, great, it's shifting gene expression back to control. We solved it. Except that when we looked at it, and I won't go through all this detail because that's a whole nother lecture that I'm not qualified to give, is that the changes in gene expression occur. So here's the control and the HFpEF model, and here's the the EMPA treatment alone, which is different from the control. So EMPA is already doing something to gene expression. And if we look carefully at what happens with the EMPA on the HFpEF model, it's not making it go back to control. It's moving it off like in a different direction. And so what we think about that is that maybe this is just instead of reverting back to a control condition, it's moving to a different good enough solution in phraseology popularized by Jim Weiss and at UCLA with the people who are at UCLA will remember the name of it. So too many words. So yeah, I'm not going to go through that. But so because I threw a lot of data at you, I'll put this slide up here, and then I'll put it back up for questions in a minute. But conclusions are hemp kinase is important, big deal. And with ROS, S-nitrosylation, glycanylation, and oxidation, it contributes to different functions and dysfunctions in ways that are specific for those different post-translational modifications. Meaning that hemp kinase is not only pleiotropic in its targets, but also pleiotropic in the things that promote its activation. And my guess is that'll also be very localization dependent in the cells where hemp kinase gets activated differently by those things. So I won't go through that, but as I said, I'll put this back up. And acutely, it inhibits myocyte late sodium current, ROS production, and hemp kinase effects that are related to EC coupling and arrhythmias, despite the fact the myocytes appear to lack SGLT2. And people have done knockouts of SGLT2 and see the same beneficial effects. We haven't done that, but we can't detect it in our gene expression. The empagliflozin, when treated, so it acutely has these beneficial effects, but chronically it can prevent the development of the HFpEF phenotype and all of these cellular consequences that influence both EC coupling and electrophysiology. So with that, I'll close with, I tried to show pictures of people who were doing this stuff as I went along, but this was a Napa Valley kind of a day out for the lab year and a half ago. Nice day there. Here's Juliana and where's Ben? Oh, there it is. And then here's when we're back at the lab and Juliana was leaving to her faculty position back in Colombia, where she was from. And she's the one who's a veterinarian and did her PhD with us and a little bit of postdoc time. So with that, I'll stop and I'll put that slide back up and have some questions.